Okay, in this second video that has to do with what DNA's job is in a cell, we are going to pick up the story that we left off with last time, and we are going to talk specifically about how that DNA code is translated into a protein. So remember, you learned that DNA contains the code for making a protein. Well, there's two steps. The first step was transcription. Transcription is copying the DNA onto this other copy, this other kind of nucleic acid, mRNA or RNA. And the second step is for the RNA to actually direct the formation of the protein. So that's what we're going to talk about this time. Now, there are three types of RNA. The one we've been talking about so far is called mRNA. mRNA stands for messenger RNA. Messenger in that it is carrying the message. It's going to carry the message of the DNA. And so it's going to be that strand that got copied from the DNA template. The second type of RNA is called tRNA. tRNA stands for transfer RNA. And transfer RNA's job is going to be to transfer amino acids to the site of protein synthesis. And we'll talk about that in just a minute. The third type of RNA is rRNA. rRNA stands for ribosomal. I apologize, I forgot how to spell. Ribosomal RNA. And it's the RNA that, together with some protein, make up the ribosome. If you recall, we talked about the ribosome earlier in the year, and the ribosome was the little kind of polka dot things inside the cell. The ribosome is the site of protein synthesis. So this is where it's all going to happen. And there is a certain kind of RNA that is makes up the ribosome. So we pick up our story where RNA has been transcribed within the nucleus. This represents here the nuclear membrane. It's been transcribed from the DNA, and it's going to leave the nucleus with the code. Remember, this is mRNA. It's going to go to a ribosome. It's going to bind to the ribosome, and it's going to begin directing the formation of a protein. This process, again, is called translation. Once it reaches the ribosome, there's actually two parts to the ribosome. There's a small subunit and a large subunit, which are separate molecules. When the mRNA arrives, it binds to the small subunit, and that's going to actually help the large subunit attach. So this whole thing is the ribosome, and the ribosome is the site where protein synthesis or where the protein will get made. And it's only happening when the mRNA is attached. Now, at the same time, remember the tRNA, the transfer RNA molecules, are going to be floating around, and they're attached to amino acids. Each tRNA molecule has a specific amino acid that it carries. That's its job. It will attach to only a specific amino acid. The tRNA's job is to carry that amino acid to the ribosome, and it's going to bring the amino acid. The tRNA will come and bind and bring the amino acid at the appropriate time following the code on the mRNA. Now you might wonder how that can happen, and I'll tell you. When the mRNA code is read to make a protein, it's read 3 nucleotides at a time. So it's kind of like that's the language of DNA. It's in threes. We've been talking about the nucleotides as A, T, C, and G, or A, U, C, and G, but they're not read one at a time. They're read 3 at a time. And every 3, it's like it's a word. Every 3 is like a word, and these are the letters of the word. So the 3 nucleotide code, we call the genetic code. So you might hear that term, the genetic code. That's what it's talking about. The 3 nucleotides on messenger RNA make up what is called a codon. Okay, so you'll have 3 nucleotides that are read at a time like a word. That's a codon. A codon is like a word, 3 bases, 3 nucleotides. On the tRNA, the transfer RNA, it also has 3 nucleotides that are read, but it's an anticodon. It's the matching base pair. So for instance, you might have a code ACU on mRNA. That would be the codon. the matching anticodon might be UGA, if that makes sense. So this would be our codon, and that would be on mRNA. The matching anticodon would be UGA here. So the anticodon will pair up with codons on the mRNA, and that's how the code is read, because each tRNA will come in, bringing its appropriate amino acid based on the anticodons, and one at a time they'll get pieced together. Now this is kind of what tRNA looks like. There's a couple different versions here. Remember, it is made of nucleotides just like DNA and any other RNA. So it's got all of your nucleotides. The way it's actually put together, it kind of has a clover leaf shape a little bit. It has this strange shape to it, and you can see the little bases. The 3 sticking out at the end here are the anticodon. So 1, 2, 3 bases sticking out. This one has, it looks like uracil, adenine, and guanine, so GAU. And then this is another way of kind of representing it in a more simple fashion. Again, you've got the 3 bases down here, the anticodon. And then at the top is a site of attachment for the amino acid, and it's different for every tRNA molecule. Now, we have worked out the entire genetic code, and we know for every combination of 3 bases or codons, we know exactly what amino acid that codes for. We know for every codon what amino acid will be brought in for that, and scientists have worked that all out. So oftentimes you'll see a chart like this that shows the genetic code. The way you read it is you have the first base in the codon here, the second base here, the third base here. So if the first base is U, you're going to read in this section. If the second base is also U, you read in this box. If the third base is U, it's going to be PHE, which stands for phenylalanine. If it's C, it will also be phenylalanine. Okay? Notice there's some redundancy in this code. That's very important, that the code actually is redundant, meaning you'll have here 4 different codons that represent ALA, which is alanine. There's 4 that represent VAL, which is valine. Arginine, again, 4. Serine, 4. Okay, so there is a lot of redundancy in the code, so that if there's a slight error, all right, if you have U and C, it doesn't really matter what that third base is. If there's an error, it will still be serine. It's kind of a way of preventing too many errors. But this is our genetic code. That's the whole genetic code. So let's talk a little bit about the process. The process begins with something called the start codon, which is always AUG. The AUG codon also codes for the amino acid methionine. So the first amino acid will be methionine, because that's the start codon. Once the tRNA molecules bring in the methionine, they're going to continue to bring in more amino acids, one at a time. They read the mRNA sequence, 1 codon at a time, so 1 word every 3 bases. And while the tRNAs are bringing in these amino acids, at the top of the tRNAs, there are enzymes that are going to bind those amino acids together to form a chain. We call that a polypeptide or a protein. And the bond that's formed is called a peptide bond. This is kind of what that looks like. So here we have our ribosome. Here's the mRNA chain, the mRNA transcript, or the copy that was from the DNA. And then here's the tRNAs coming in. We have a sequence here. Here's an amino acid. Here's an amino acid. Here's an amino acid. Here's the tRNAs coming in. Here's your codons, anticodons. So it'll come in with the proper amino acid. Up here, the peptide bond will be forming. Now this will continue until it reaches a particular codon called a stop codon. You may have noticed on the genetic code a few slides back that there were some that said stop. There are, I think, 3 different stop codons, and that's the signal. When it reaches that codon, there is no tRNA molecule that matches that. There's no anticodon to these stop codons, so no more tRNA molecules will come in at all. And that signals the end of the protein. At that point, the finished protein is detached. The ribosome comes apart. the finished protein is detached, and it's going to float away to the ER where it is processed. If you recall, the endoplasmic reticulum's job is to process proteins, and so that protein already is there at the ER, and it's going to begin to be processed. So here's an overall view, a review of the process of protein synthesis. It begins when DNA is copied onto RNA, and it's messenger RNA. Messenger RNA is going to attach to the ribosome. Okay, so that's happening here. Here's our ribosome. The next thing that happens is the tRNA molecules bring in the amino acids one at a time. Okay, and finally the amino acids are bound together, bond to form a protein. Okay, so that's a lot to learn all at once. Some of you may have heard some of this before, but I'm sure it's still kind of a lot. We're going to spend some time in class processing all of this and making sure that you understand it. But that's it for that, and we'll see you later.